and one with double bonds in it. Okay? So that's the melting point thing there. Yeah?
We have to know if we got like trans versus cis.
The only thing you do need to know is that naturally occurring unsaturated fatty acids are in the cis double bond form. Trans fats are made through like industrial processes and actually are not naturally occurring unsaturated fatty acids. So the naturally occurring unsaturated fatty acids are in the cis configuration. Question? Oh, you're good? Okay. All right. Let me make sure that was it for fatty acids. Okay. Some specifics after that are it turns out some of your unsaturated fatty acids are also classified as essential fatty acids. What an essential fatty acid is one that the body cannot produce itself. So they get it through so in order to get essential fatty acids, you get it through your diet. The 3 essential fatty acids that you have to know by name are listed right here. These are all unsaturated fatty acids: linoleic acid, alpha linoleic acid, and gamma linoleic acid. So it turns out if it has the word linoleic in it, it is going to be classified as an essential fatty acid for the purposes of this class. So again, what an essential fatty acid is one that that we cannot make. We have to get it through our diet. These are all essential in the production of higher ordered fatty acids, of ones with a lot more carbons in them. They give you 2 examples. Arachidonic acid is another important unsaturated fatty acid that we make out of linoleic acids. And this really long, complicated name, DHA, docosahexaenoic acid, is also a component of linoleic acid. You see these numbers here, 18, 2, 3s, and stuff like that. You don't have to memorize these numbers, but if you are given an unsaturated fatty acid with these numbers written in, you should be able to know what they mean. So you don't have to memorize these numbers, but if you see these numbers on the exam, you should know what they mean. And here's the following. There are 3 numbers to worry about. One is the first number written here. In this case, it's 18. This number tells you that the fatty acid you are looking at has 18 carbons in it. So the first number here tells you how many carbons are in them. The second number tells you how many double bonds are in them. So linoleic acid is composed of 18 carbons and composed of 2 double bonds somewhere in there. The last number is called the omega number. In this case, omega 6. So here's what the omega number is. Let me find a picture of linoleic acid. It's not a good picture, so let me zoom in a lot. Okay. So the first number tells you how many carbons are in there. The second number tells you how many double bonds are in there. The third number is called the omega number. It tells you the following. The number of carbons from the end you have to count until you hit the double bond. So let's count for linoleic acid. Carbon 1, 2, 3, 4, 5, 6. We're not numbering the carbons, we're just counting how many carbons we have to travel before we hit the double bond. It's a poor picture here. It'll be a clearer picture on the exam, but this is the double bond for linoleic acid. You don't have to memorize that it's omega 6. You may be given an unsaturated fatty acid you've never seen before, but you have to know what the omega number means. In this case, 1, 2, 3, 4, 5, 6. This means that this fatty acid has an omega number of 6. What if I were to ask you what the omega number is for alpha linoleic acid? 3. 1, 2, 3. Done. That is the omega number for alpha linoleic acid. Okay? So that's so again, the most important part are if you're given a fatty acid you've never seen before and it comes with these numbers, you have to know what the numbers mean. First number tells you number of carbons. Second one tells you number of double bonds. Third number is a way to tell you where the first double bond is when you count from the end. That is called the omega number. Okay. All right, moving right along. And I will go over this more in detail at SI. The second possible extra credit question. And this one looks more complicated, but in my opinion is a little bit more straightforward to explain. Has to do with what's called the inflammation pathway, the inflammation cascade. Annoyingly, it is split up into a couple of different places in Chapter 8a, so I will jump straight to the most important slides, and I will go back and talk about this stuff that I'm skipping. Yeah, this is probably the most important slide when you are talking about inflammation. So one possible extra credit question will say, describe the inflammation cascade, the components of it, and how inhibiting the components of it leads to certain side effects. So you guys know aspirin and ibuprofen, those are your inhibitors of pain receptors, right? It's more, in fact, it doesn't actually inhibit pain receptors. Those drugs are pain relievers because they actually inhibit these enzymes here called cyclooxygenase 1 and cyclooxygenase 2, COX-1 and COX-2. So, how to get full credit on extra credit if this ends up being your extra credit question? Describing the inflammatory cascade. Basically, describe everything written here on the left. You don't have to draw any of these. Please don't, because I don't know what they look like. I just want you to tell me that the inflammatory cascade is basically the following. You take arachidonic acid, and you can even tell me where it came from. It is transformed into two categories of molecules, and that's the one thing they don't write out here. But they take arachidonic acid and COX-1 and COX-2, transform it and turn it into one, a class of molecules called prostaglandins, or two, a class of molecules called leukotrienes, which might be on the next slide. Yeah, right here. Let me zoom in. Wherever you see the phrase PG something something, that is a prostaglandin. Again, don't draw all this. I don't know what this stuff looks like. You could draw whatever you want and I'll probably believe you. What you need to show me is that the inflammatory cascade is conversion of arachidonic acid into inflammatory molecules, leukotrienes, and prostaglandins. Okay? So just the names of the molecules are fine. I don't even mean the specific names. I mean the category, leukotriene, prostaglandin. The reason what these molecules do is they actually cause the symptoms of inflammation. Redness, swelling, increased temperature. These molecules cause that. So, we have several drugs to inhibit that. And the drugs that are listed are down here: aspirin, ibuprofen, Vioxx, and Celebrex, or whatever it's pronounced. Each one of these either inhibits both COX enzymes or one of them. The last bit of information you have to tell me, after telling me what the molecules are converted into, what inflammation cascade means, what drugs inhibit these enzymes, the last thing you need to tell me are the possible side effects of inhibiting these enzymes. And all that information is listed here. Inhibition of COX-1 is known to produce renal toxicity and stomach bleeding. In fact, a lot of older versions of aspirin, and even now, if you take a lot of aspirin, you're going to run into these side effects. So again, inhibition of COX-1 produces renal toxicity, which is kidney problems, and stomach bleeding. Inhibition of COX-2 is actually known, in some cases, to cause heart attacks in extreme cases. So you need to tell me the potential side effects there. All right? So I'm not going to draw all this like I did for glycogen phosphorylase, because glycogen phosphorylase is simple enough that you should be able to draw it. For this, I need either bullet point explanations or paragraph explanation. Same reasoning as with glycogen phosphorylase. Show me that you know enough about this that you weren't cheating. That's about it, and I will give you full credit on that. Okay? So I just wanted to mention that as your other possible extra credit question. This topic does not usually show up as multiple choice questions. So the most that you have to know it is to be able to explain it in a written form, and then you're fine.
We don't need to know which drugs do which, right?
No, just tell me that these drugs inhibit COX enzymes. I'm going to be very lenient this semester. Okay. All right, triglycerides. So, jumping back, after fatty acids, The next category of lipids is called triglycerides. And the reason why they talk about this is that it turns out fatty acids do not naturally exist by them floating around by themselves. Fatty acids are, in nature, are associated most commonly to a glycerol molecule. So the phrase that they use to describe fatty acids attached to a glycerol molecule, to describe 3 fatty acids attached to a glycerol molecule, is the phrase triglyceride. And you may have heard triglycerides in other contexts regarding health. Triglycerides are your energy storage molecule that lipids take, for lipids. The energy storage molecule for sugars, you learned, was glycogen and starch, right? The energy storage molecule for lipids is triglycerides. Fat cells, also known as, and I may mispronounce this, adipocytes, something like that. You guys know, you guys take your health-related classes. I don't study that stuff. I study the eye a lot. But adipocytes, which are fat cells, have lots of triglycerides in them, okay? That's all you need to know about triglycerides, is that they're the energy storage molecule for lipids, they have fatty acids in them, and you find them in fat cells. That's about it. The next category of lipids, I will jump to this picture. The next category of lipids is called glycerol phospholipids, okay? So glycerophospholipids, again, you don't have to memorize each specific one on here. You may have to for the final, and if the time comes, I will talk about it. But if given any one of these, you need to tell me it's a glycerophospholipid. So how do you identify glycerophospholipid? There are 2 fatty acids attached to it instead of 3. Triglycerides had 3 attached. Glycerophospholipids, there are 2 fatty acids attached to it. A glycerol, which are these 3 carbons here in the middle. A phosphate, and some weird looking functional group series at the front end here. Okay? Doesn't matter what it is. They give you a few examples. This stuff right here, let me zoom in. This stuff right here is choline. This stuff is ethanolamine. This is serine, which actually probably looks like serine. And they give you a bunch of examples. Doesn't matter. The most important thing is if they give you any one of these structures to tell me, via multiple choice answer, that it is a glycerophospholipid. So again, look for 2 fatty acid tails, a glycerol here, and a phosphate head, a phosphate plus something special attached to it. And that's a glycerophospholipid. So, we went over fatty acids, we went over triglycerides, we went over glycerophospholipids, so you're just looking for short, quick identifiers to be able to distinguish them from each other. This was the inflammation cascade. I'm going to skip down to the next category. The next category is ether glycerophospholipids. And even amongst this, the only thing you need to know is that the one example of ether glycerophospholipids is plasmalogens. You don't even have to memorize the picture of plasmalogens. It's here, I think this is, no, that's not plasmalogen. Where is plasmalogen? There isn't even a picture for it. This is just a picture of a general ether glycerophospholipid. To identify an ether glycerophospholipid is very easy. You just identify a glycerophospholipid like we did earlier, and then you look very carefully at your fatty acids. If one of them is an ether link instead of an ester link, you know you have an ether glycerophospholipid. That's it. The only specific ether glycerophospholipid that is listed in the notes, and therefore the only one that is important to know by name, is plasmalogen. So if you see this phrase, if you see that vocab word, know that it is an ether glycerophospholipid.
Those still have the 2 fatty acid tails?
Yep. Every other feature with glycerophospholipid is the same. There's just one difference, is that one of the fatty acid tails is connected via ether linkage rather than an ester linkage. For comparison's sake, let me go back to glycerophospholipids. Notice both tails are ester linkages. So ether glycerophospholipids, one of them turns into an ether linkage. That's it. I don't know if it's here. Somewhere it's in here, but I can't find it right now. But plasmalogen, biologically, you need to know that you find this in the cell membranes of heart cells. So write that down. That plasmalogen, we find it in the cell membrane of heart cells. I can't remember where it is on here, but that is a unusually specific biological fact that sometimes shows up on exams. That plasmalogens are ether glycerol phospholipids that you find in the cell membrane of heart cells. All right, now we get into the next category of lipids, which is sphingolipids. So, sphingolipids are lipids that have the following base shape, and then they attach a bunch of stuff to it. Sphingolipids are based off this structure here on the left called a sphingosine. You can compare this sphingosine to all the other lipids that we looked at. A sphingosine does not look anything like a fatty acid, does not look anything like a glycerol phospholipid or an ether glycerol phospholipid or a triglyceride. It's its own thing. When you see a sphingosine, and by extension a ceramide, which is a modified sphingosine, that is a sphingolipid. Now, sphingolipid, there are 3 actual types. You take a ceramide, like this on the right here, and you do one of 3 things. So here's the ceramide down here. If you attach a phosphate head to a ceramide, you call it sphingomyelin. Okay, and again, I'll go over detail of this on Thursday during the SI session, but I'm just giving you some short, quick hints on how to identify sphingolipids. Sphingomyelin is you take a ceramide and you attach a phosphate head to it. You might even recognize that these are some possible phosphate heads that you saw in glycerol phospholipids, but in this context, they're attached to a ceramide here. If instead of a phosphate head, your ceramide is attached to a single sugar, they call that a cerebroside. Okay? If your ceramide is attached to a single sugar, that is called a cerebroside. The next structure, in my opinion, is the easiest structure you will ever identify in this class. And it looks complicated, but I bet nothing else in this class looks like this. Is that there is a ceramide attached to a bunch of sugars that is a ganglioside. A gang of sugars is how I memorize it. Okay? It's as easy as that. This, in Biochem 1 or 2, will be the easiest thing to identify because nothing else looks like this. A ceramide attached to a bunch of sugar, into a bunch of sugars is a ganglioside. Okay. Don't worry about lipid storage diseases for the exam. If you need it for the final, I will give it to you then. Don't worry about waxes either, or terpenes, or even more terpenes. The last category of lipids to worry about are what is called steroid hormones. And they all look very similar to each other. So again, you don't have to identify each individual steroid hormone, but if you're given something that looks like any one of these, just tell me that it is a steroid hormone in the multiple choice question. So a steroid hormone is another category of lipid that is derived from cholesterol. That it took a cholesterol structure, which is this 4-ring structure here, and modified it into various different possible steroid hormones. Some of these you might recognize are sex hormones: testosterone, progesterone. Sometimes there's a couple other ones they give you, but overall, if you see any of these shapes, they are called steroid hormones. Sex hormones are a specific type of steroid hormone, so be very careful of that. If you see just any one of these categories, the more accurate statement is to say they are steroid hormones, because in this class you won't be asked to identify anything more specific than the category. So the category of these things are steroid hormones. Okay. So that's it for 8A. Let me make sure I didn't forget anything. And that's it. You may be relieved to see a lot of 8A is just looking at little specific features and distinguishing the different lipids from each other, and that's it. You don't have to memorize really complicated lipid names, just the categories. So, 8B, yep, that's on the exam too. And again, feel free, if you need to go, you can.
Will you review this?
I will try to do as much of 8 as possible on the Thursday SI session, but I'm giving you guys the most important parts for chapters 8A and 8B. All right. So, first we will begin with this picture. Chapter 8B talks about cell membranes. what is the composition of cell membranes, how cell membrane pumps work, how diffusion works, how active transport works, passive transport works, all of that. The very first thing to talk about in terms of cell membranes is what type of proteins you get in a cell membrane. And it's simply the following. You either get integral membrane proteins, which are membrane proteins that are fully embedded into the cell membrane. This blue thing right here is an integral membrane protein. Notice it is driven straight through the cell membrane. And you also get peripheral membrane proteins. Peripheral membrane proteins are just loosely associated with either side of the cell membrane. They're loosely associated. They can attach and detach. Okay? So integral membrane proteins are embedded. Peripheral membrane proteins are loosely associated.
How do you know the green one's like integral? Because it looks like it's peripheral.
It would be, they would illustrate like an interior part of it. So the only one, as far as I can tell, is this pink one, this purple one, and this yellow one seems to be embedded in there. In this one, although it says integral, on the exam it'll be very obvious if it's an integral one or a peripheral one. So this is the only picture though they have in the notes here. But on the exam it'll be very obvious if the protein you're looking at is integral or peripheral. We've already seen this picture here. It's a reminder of how a cell membrane is formed. Again, a cell membrane is formed from phospholipids, from two-tailed phospholipids, right? And they're held together by hydrophobic forces. So a cell membrane is held together by hydrophobic forces. That's the forces keeping these long nonpolar tails together. These hydrophobic ends of these amphiphilic molecules are held together via hydrophobic forces. They give you plenty of pictures, but you don't really need the specific pictures anymore. But yeah, this is just another diagram illustrating integral membrane proteins versus peripheral. Don't worry about the many complicated names of gated channels or anything like that. This next series of information talks about lipid anchors. You don't need to know all the different complicated lipid anchors. The only thing you need to know of what a lipid anchor is a thing used to keep a peripheral protein attached to the cell membrane. Okay? So what a lipid anchor is a special thing, is a special anchor, it's in the name, special lipid component that keeps peripheral proteins at least temporarily attached to the cell membrane. So they are attached via lipid anchors. Integral membrane proteins don't need this. Integral membrane proteins are driven straight through the cell membrane, so they don't need lipid anchors. But peripheral membrane proteins are attached via lipid anchors to the cell membrane. So there's a bunch of different types, so I'm going to skip over them because you don't need them. And we'll go into testing. And we'll go into the different ways that molecules are transported through the cell membrane. We'll start with the simplest one, passive diffusion. Passive diffusion, by definition, is a type of transport that doesn't involve any channel proteins or carrier proteins. All passive diffusion is the physics behind something that is highly concentrated moving to an area that is less concentrated. So, in this example, the highly concentrated molecules over here are going to diffuse through the cell membrane to the side that is less concentrated. That is all passive diffusion is. It doesn't require any ATP. It is a natural mechanism that exists simply due to the nature of physics, where high concentrated stuff moves to low concentrated stuff. Oh, certain molecules can more easily diffuse through the cell membrane though, right? And think about it. What type of molecules do you think will more easily diffuse through a cell membrane? Polar molecules will have the hardest time to diffuse through a cell membrane, right? Because the cell membrane has all those non-polar hydrophobic tails. A polar molecule will not be able to pass through easily. But a non-polar molecule, such as a steroid hormone, or a lot of those lipids are non-polar, they're able to diffuse through the cell membrane very easily because of all that non-polar property, hydrophobic property that lipids have. So polar molecules don't easily diffuse through the cell membrane. Non-polar molecules very easily diffuse through the cell membrane. Same process with charged particles. Oh, I guess this is where he talks about it. you will get, and this is special for this semester, brand new, you will get a calculation question that is based off this exact formula right here. Okay? So basically, the question will give you a concentration of an ion outside the cell and inside the cell. It will give you two different concentrations, and it will ask you, what is the electric potential generated by this difference in concentration? You have all the numbers here. 61 millivolts times the log of the concentration of whatever ion is given on the outside of the cell divided by the concentration of whatever ion is given on the inside of the cell. So this is the exact formula. Okay? It's a simplified version of what is called the Nernst equation, which you actually learn about in more detail in Biochem 2, but the MCAT is starting to put some like Nernst equation type questions on it, so this is why it's talked about here. So again, this formula is used to calculate the electric potential generated due to a difference in concentration of ions on one side of a cell membrane versus another. All you'll have to do is, if you're given the concentration of the ion outside the cell membrane and the concentration of the ion inside the cell membrane, plug it into this formula, calculate the electric potential. So really the only number you have to memorize is 61 times log of ion out divided by ion in. I'm not going to go over an example because it's a pretty straightforward formula. Just plug in the numbers and you get your answer. Okay? Yes?
Are we not doing units?
No, the units actually will cancel out each other because it's concentration in the numerator and the denominator. Yes?
Can we bring calculators, or do we have to memorize?
Yep, make sure you have your scientific calculators, because not only do you need the scientific calculator for this, you will need it for enzyme genetics as well. So make sure you bring a scientific calculator. Alright. Nope. Nope. Alright. 61 is a constant at 37 degrees Celsius. So we actually simplified the Nernst equation for you. So that's why, yes, memorize the constant of 61 millivolts for that equation. Okay. Now getting back to more comfortable things. After passive diffusion, there's another type of transport of molecules across a cell membrane called facilitated diffusion. Facilitated diffusion is very similar to passive diffusion, but one difference, and hopefully there's a picture. Nope. Nope. No, there is no picture of facilitated diffusion, so I'll just have to describe it. So facilitated diffusion is passive diffusion with the help of channel proteins in the membrane. So what this means is there are channel proteins, openings in the cell membrane specific for certain molecules. They will allow certain molecules to pass through if that molecule binds to the opening in the cell membrane, to that channel protein. Facilitated diffusion is the same thing as passive diffusion, just with the help of channel proteins. Molecules still flow from high concentration down to low concentration, just like physics wants to happen. It'll just do it through channel proteins. That is what facilitated diffusion is. So another way of saying it goes from high concentration to low concentration is saying that it flows with the concentration gradient. That molecules flow with the concentration gradient. Okay. Blah, blah, blah. Other terms to note. This applies to either passive or active transport. If you see the phrase uniport, all that tells you is that this channel protein takes one molecule and transports it in one direction. If you see the phrase symport, it means that channel protein takes two molecules at a time, two different molecules at a time, and transports them in the same direction. If you see the phrase antiport, it means that the channel protein takes two molecules at a time and transports them in opposite directions simultaneously. Okay, so if you see any of these words at any point in 8B, in the 8B PowerPoints, that's what they mean. Let me get to a couple things. So, they give you a couple examples of cell membrane transporters. The very first example they give you is an example of facilitated diffusion, not active transport. It's an example of facilitated diffusion. If you see the phrase, the transporter is called sodium glucose transporter. That is a symport, a facilitated diffusion that is a symport. What this means, what this transporter does, is you find it in the small intestine. When glucose and sodium ions simultaneously bind to this transporter. It transports both of them into the cell membrane. So when you eat some food and there's some salt associated with it, when both the glucose and the sodium ions bind to this transporter, the small intestine absorbs that glucose and that sodium into the small intestinal cell, and then eventually that gets transported into the bloodstream. You used to have to know really specific details about this. For this semester, you don't need the really specific details. Just know that the sodium glucose transporter works as a symport. It takes two molecules and they transport them in the same direction. And also, and I believe you talked about this at least somewhat in class, there's a special drug that exists to block this transporter, okay, called, I think it's Farxiga or something, I don't know how it's pronounced, but if you see this drug here, all you have to know is that it inhibits the sodium glucose transporter. That's it. Okay. Now for the most important pump in the whole 8B, and in probably all of biology, is the sodium potassium pump. Alright, so I'm going to draw this one out actually. Okay, so
You're so tricky.
okay. So the sodium potassium pump is an example of active transport. In fact, the full name of the sodium potassium pump includes the phrase ATPase. The reason why that is what this pump does, and all active transport pumps do, is they pump molecules against the concentration gradient, meaning they take molecules on the low concentration side and they push them to the high concentration side, which goes against what physics wants. That requires energy. So active transport pumps require energy to pump molecules against their concentration gradients. That energy comes from ATP. They use ATP in order to function. Sodium glucose transporter was not an active transporter pump, so that one didn't require ATP. It was passive transport. This one is active transport, so it requires ATP. So, the main feature of the sodium potassium pump is it takes 3 sodium ions. They bind to the intracellular side of this pump. So what I mean by intracellular side is this pump has an outside portion and an inside portion, okay? And the very first thing that this pump does is it grabs 3 sodium ions and it binds to the inside portion, getting ready to be shipped out of the cell, okay? That's the first thing that happens. One is that 3 sodium ions bind to the inside portion. The second thing that happens, and it kind of happens simultaneously, is that ATP binds to the inside portion of this pump. So everything is getting ready. It is getting ready to pump the sodiums out of the cell. That is what this pump does.
Where's ATP going? I'm sorry.
ATP also binds to the inside portion of this pump, the intracellular portion of the pump. So 3 sodium ions, an ATP molecule binds to it. Then what happens is ATP is hydrolyzed to activate this pump. ATP is hydrolyzed into ADP and activates this pump. It sends 3 sodiums out of the pump and opens up the outer portion of the pump. Okay, so it just threw out 3 sodiums. And now we have, what's that?
Sending out 3 sodiums, is that step 2 still?
The 3 sodiums is still step 1. Yeah, so
Sending it out.
yes, the sending it out is step 2, yes. Is sending the 3 sodiums out is step 2. Okay? After the 3 sodiums are out, and after the pump has been exposed to the outer portion of the environment, 2 potassium ions are going to bind to the outer portion of this pump. Okay? The next step, I guess I should write that down. 2 potassiums bind. Okay? The next step is that ADP is removed. And for some reason, there's something in my head that says I'm probably forgetting something. So tomorrow I'll cover this again. But right now, the next step is that ADP is removed and the 2 potassiums are pumped into the cell. Okay. And when the 2 potassiums are pumped into the cell, you're ready to begin to go back to the beginning. Is that after 2 potassium are pumped into the cell, you can take 3 more sodiums and bind to the intracellular portion of the pump, and brand new ATP to bind to the intracellular portion of the pump, and you begin everything again. So once the 2 potassiums are pumped into the cell, you start over. Okay. So this series of steps is repeated over and over again. So you're continuously pumping sodium out of the cell and potassium into the cell. Which side of the cell has a higher concentration of sodium ions? The outside, right? And that is because, and you can figure that out by reminding yourself that the sodium-potassium pump pumps against the concentration gradient. It is taking sodium ions inside the cell and pumping it to the side that already has a lot of sodium ions in it. So it is going against the concentration gradient. So the extracellular concentration of sodium is higher than the intracellular concentration. Yes?
Is the intracellular concentration of potassium higher than the outside?
Exactly. The next thing is to note that potassium is also being pumped against its concentration gradient. The intracellular concentration of potassium is higher than the extracellular concentration of potassium. So both of these things are going against their concentration gradient. 1 second. Let's see, do that. Oh yeah, is this a uniport, symport, or antiport?
Antiport.
It is antiport because these molecules are going in opposite directions. Almost done. All right. One more pump is important. Yes,
Do
question.
you have to know about the 3:2 ratio for the sodium potassium?
You do have to memorize the numbers, yes. It's 3 sodiums and 2 potassium, yes. All right. Is this it? All right. One more pump that you have to know about, and then that actually is it, is this one here. This one is called hydrogen potassium gastric ATPase, or the hydrogen potassium pump. You see the phrase ATPase? This is an active transport pump. It is pumping ions against their concentration gradient. This is a special pump in that it is actually composed of 2 different pumps put together, which I will show you over here on the right. The goal of the sodium potassium pump is you find this pump in stomach cells. Remember that the stomach environment is a very acidic environment. Well, how does it become acidic in the first place? It turns out that your stomach cells have all these pumps called the hydrogen potassium pump. Its net job is to pump hydrochloric acid out into the stomach environment to make it a very acidic environment. It does so via a 2-step process. Step 1 you see up here is an antiport between hydrogen and potassium. Step 2 is down here. What type of port is this? It is a symport. You are taking potassium and chlorine, coupling them, and transporting them in the same direction outside the cell. So this is kind of a unique pump in that although the name is hydrogen potassium pump, it is actually technically 2 things put together. The main hydrogen potassium pump up here, which is an antiport that absorbs potassium and ships out hydrogen. And this secondary component here, where the potassium inside the cell couples to chlorine and is shipped out of the stomach cell into the stomach environment. Notice that potassium goes in a cycle of inside and outside, so it's as if potassium doesn't really change at all. The only net difference is that hydrogen is being pumped out and chlorine is being pumped out of the stomach cells into the stomach environment to create a very acidic environment. Okay, so very important to note that the hydrogen potassium pump is actually two different pumps, an antiport and a symport.